Assalamualaikum to our lecturer Dr. Shazila and our fellow friends. We are from Group 2. We'll be discussing about a fungal toxin which is called Aflatoxin. And the next slide will be showing our group members, and the topic that will be discussed in this video will be the production process, origin and symptoms of aflatoxins, how to prevent it, and some case study that related to aflatoxins.
Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Anisa binti Edi Zulfa, matrix number 1201002, and now I shall explain on the production process of aflatoxin. Aflatoxin is a toxin mainly produced from Aspergillus flavus and also Aspergillus parasiticus. There are 4 common types of aflatoxin, which includes aflatoxin B1, B2, G1, and G2. So, Aspergillus flavus commonly produces B-aflatoxins and also mycotoxin, cyclopyrazonic acid, CPA. Meanwhile, Aspergillus parasiticus produces B and G aflatoxins. Between the two, Aspergillus flavus is the most widely reported foodborne fungus, and this fungus is commonly found abundant in the tropics. The production of aflatoxin involves the polyketide pathway, which consists of 27 enzymatic reactions, making it a very long and complex reaction. After this pathway occurs, this produces the aflatoxin, which includes aflatoxin B1, which commonly intoxicates mammals. So after aflatoxin B1 is ingested, this is then metabolized in the body of mammals by the liver and produces an epoxide intermediate, which is carcinogenic to mammals, or a hydroxylated aflatoxin M1 and M2, which is less harmful to the body. Here are some of the factors that influences the production of aflatoxin in fungi. This includes the temperature, nutrient sources, light exposure, water activity, pH, oxidative stress, and also other genetic factors.
My name is Fatin Afika, and next I will cover the part of the history of aflatoxins. The history of aflatoxins began in the early 1960s when they were discovered in England due to 100,000 turkeys dying from a disease known as turkey eggs after being fed with contaminated Brazilian groundnut meal on a poultry farm in London. A veterinary expert named William Percy Blount conducted extensive lab study on the turkey outbreak of egg sickness, and he concluded during his investigation that aflatoxins from a mold named Aspergillus flavus in Brazilian groundnut meal had caused the outbreak. Next, we move to the source of aflatoxins. Aflatoxins can contaminate some crops including cereals like maize, sorghum, rice, and wheat, and for oilseeds such as peanut, sunflower, soybean, and cotton, followed by spices including chili peppers, black pepper, coriander, turmeric, and also ginger. And for the tree nuts, the example is almond, coconuts, Brazil nuts, walnuts, and also pistachio. Other than in plants, aflatoxins also can be found in the milk, eggs, and meat from animals that fed with contaminated feed. When someone is infected with aflatoxins, they will show some of the symptoms like loss of appetite, edema, vomiting, abdominal pain, liver necrosis, coma, and worstly, death. Meanwhile, animals also will show the symptoms when they are exposed to aflatoxins, and some of the symptoms are similar with the human symptoms, such as sluggishness, loss of appetite, vomiting, jaundice, liver damage, unexplained bleeding, and also diarrhea. When it comes to the aflatoxins, usually this mold will attack at the liver and causes serious liver damage and also liver diseases.
The implication of aflatoxin and their potential threat to humans have clearly shown a need to eliminate or at least minimize its contamination in food and feedstuff. So there are few methods that can be applied to prevent the issues, which are detection and screening, pre-harvest control, post-harvest control, biological control, and breeding of crop that can resistant to fungal growth. For pre-harvest control, aflatoxin contamination can be reduced somehow by appropriate culture practices such as irrigation and control of insect pests prior to harvest. As for post-harvest control, significant emphasis has been placed on improving storage conditions that are less favorable for fungal growth. For detection and screening, surveillance program has been established to reduce the risk of aflatoxin contamination in human and animal. Some techniques include TLC, HPLC, GC, RIA, and ELISA are available for rapid detection of aflatoxin as low as 1/10 of nanogram per milliliter. For biological control, application of non-aflatoxigenic biocompetitive native A-flavor strain to outcompete toxigenic isolates in the field has been effective in significantly reducing aflatoxin contamination. Last is breeding for commercial crops that are resistant to fungal growth. This method is the most economic and effective strategy for reducing and eliminating aflatoxin contamination by breed commercial crop varieties that show resistance to fungal infection or aflatoxin formation.
An overview of the aflatoxin tragedies. The most severe aflatoxin outbreak occurred in Kenya in 2004. The patient presented with abdominal pain, pulmonary edema, and liver necrosis after ingest 50 mg of aflatoxin B1 per day and were diagnosed with aflatoxicosis. The source of aflatoxin was attributed primarily to local aflatoxin contaminated mix. Post-outbreak investigation identified several factors that caused the outbreak. Firstly, the mist produced in the imported to Kenya was stored in warm and humid environment, which facilitated fungal growth. Secondly, no proper storage sites, and most farmers store the maize in their home with leaking roof. Next, we move to the potential solution. So, from what happened in Kenya on 2004, there were a few potential solutions that could be taken to prevent the future outbreak. So, firstly, for the governments itself, they can establish a strict regulation in detecting and removing aflatoxin contaminated crops by developing an agricultural policy. Next, they can conduct a seminars to report current harvest condition. They also can make a collaboration with researchers that could provide opportunities to battle against aflatoxin infections. Secondly, from the public health improvements, they can increase health education of local citizens. They also can make a long-term monitoring of aflatoxin levels through blood tests that can provide active surveillance to detect outbreaks early, and then also can emphasize on routine healthcare checkups can detect signs of early aflatoxicosis. And next, the local public health authorities can conduct the educational campaign on how to identify the aflatoxin contaminated crops. So these are the two types of agricultural innovation that has been built in preventing the future outbreaks. So the first one is Easy Dry M500, which is a portable dryer that helps to reduce the mist humidity because proper drying and storage condition can directly reduce the risk of aflatoxin outbreak. And next is Aflasif. Aflasif is a biological control product that can be applied to the maize field uh, to reduce the aflatoxin contamination. So from findings, it said that aflatoxin has reduced 80% to 90% of aflatoxin contamination. In conclusion, the outbreak that happened in Kenya in 2004 has revealed many problems, especially in public health management as well as agricultural procedure in the processing and storage of crops. And the short-term exposure to high-dose aflatoxin can cause acute hepatitis, while long-term exposure can induce to the liver cancer, and the development of Alpha-SIM and EasyTry M500 has led to safer processing and storage conditions for the crops.